All right, hello everyone. Um, last group of organic molecules critical for living things are the nucleic acids. All right, so what are nucleic acids? All right, so you've all heard of DNA before. DNA are very large organic molecules, and no doubt you've heard about DNA. Those are the molecules that are responsible for people passing along their traits from one person to another. Yes, that's where your genes are located on these molecules. These are information molecules that are present inside your cells. So DNA is a type of nucleic acid, and then there's another type, RNA, and we'll talk a little bit about what the differences are between those two. So your nucleic acids, just like your other organic molecules, carbon, hydrogen, oxygen, they also have nitrogen and they also have phosphorus in them. Just like your other organic molecules, these are polymers, so they're built from chains. And in this case, our building blocks are called nucleotides. And then as we're going to see, a nucleotide itself has smaller parts. Okay, and those are called a nitrogen-containing base, a pentose sugar, and a phosphate group. Okay, so these three things are the smaller parts of a nucleotide. Nucleotides are then linked together in chains to make RNA or DNA molecules. So I know lots of new terminology here as well. Functions. We're going to hear more about DNA and genes a little bit later as we move further into the semester when we get to Unit 2. But DNA molecules are where your genes are located. So when you hear about genes, they really are just parts of organic molecules called DNA, and which is pretty interesting. And these DNA molecules, they function as information storage molecules, even though they're chemicals. Genes are, as we'll see later, genes, interestingly enough, function as chemical recipes that tell your cells how to make proteins. So kind of get used to thinking about that. Genes are blueprints, recipes that are going to tell your cells, provide instructions for how to make proteins. Now, we already said proteins have lots of important jobs. They are responsible for the functions of your cells. They give your cells the traits that they possess. So genes and proteins are very intimately linked, because if genes are the recipes that tell your cells how to make certain kinds of proteins, if you pass those genes from one cell to another or one individual to another when we reproduce, then the new cells will be able to make those same proteins because they have the recipe books for doing so. I know that's pretty abstract for right now. We'll hear more about it later, and then you'll hear even more about it when you take microbiology. RNA, what's the difference between DNA and RNA? We'll see some differences shortly. And the RNA molecules help out with this sort of transition, this reading of the genetic recipes, where you're going from these DNA instructions, and cells are using that to build proteins. They help out with that process. And don't worry about that right now, because you're going to hear more about it when we get over to Unit 2, and even more about it when you take your microbiology class. All right, so what is DNA? DNA stands for, you may have heard of this before, deoxyribonucleic acid, and the abbreviation, much easier to say and remember, is DNA. DNA molecules have 4 what are called bases in them: adenine, guanine, cytosine, and thymine, which are abbreviated A, G, C, and T. We'll see more about that in just a second. And unfortunately, in biology, sometimes you see the same words used in different ways. So I know we were using base to refer to compounds that raise the pH of their environments, and here we're using it in a somewhat different way to refer to the parts of a DNA molecule. Now these parts are bases, but don't think of them in exactly the same sense as what we were talking about when we went over the pH lecture. DNA molecules have they're double-stranded, and we'll see what I mean by that, and they're helical. So remember on the protein lecture we talked about something that's helical in shape. That means spiral. DNA molecules look like spiral staircases, and you've seen pictures before, diagrams of DNA molecules, even if you didn't realize it. And again, DNA, one of its main functions, they're information storage molecules, and they contain the recipes that will tell your cells how to make particular proteins. Proteins are ultimate. There are chemical workhorses, so if cells are going to function, they have to be able to build their protein. So therefore, before a cell divides, one cell is going to split apart and become two cells. You got your DNA in here. That DNA has to get copied to make sure both daughter cells have the DNA. Because if you don't have those blueprints that tell a cell how to make its proteins, the cell's not going to be able to function. All right, here's a closer look at what a DNA molecule looks like. It's a diagram from your textbook. All right, so on the last slide we said DNA is double-stranded. So what do we mean by double-stranded? Double means two, and a strand here, a nucleic acid strand, is a chain of nucleotides linked together. All right, and then here is an example nucleotide right here. Okay, now earlier I said a nucleotide itself can be divided up into three parts. There is a part called the nitrogen-containing base, or the nitrogenous base. Okay, that's this portion of the nucleotide. And on the last slide I mentioned that in DNA, that portion in a DNA molecule could be adenine, which is abbreviated with an A, guanine is abbreviated with a G, cytosine is abbreviated with C, and thymine is abbreviated with T. So that's what that looks like. That is covalently bonded to a sugar, and this sugar is called deoxyribose. That's a pentose, so that sugar has 5 carbons in it. And then that sugar is bonded to another part of the molecule over here, and that's called a phosphate. A phosphate has a phosphorus in the center, and it's surrounded by 4 oxygen molecules. Okay, so everything that I was just highlighting for you there, those 3 parts together make up a single nucleotide. Okay, and then a strand of DNA consists of a chain of these nucleotides. So if we kind of look at this diagram over here, all right, so if you follow along in purple right there, well, it's not really showing up too well, like a thicker. You kind of follow along in purple there, that's representing one strand of DNA. And the really important, super important parts, or the unique features of DNA molecules, it really gets down to the order or the sequence of those bases, these A's and T's and C's and G's. So that's the part on the molecule that's always highlighted. So if you notice that strand of DNA that I just highlighted for you, there's a adenine nucleotide there, there's a C nucleotide, cytosine there, and then there's an A, then there's another A, a G, I hit it, but there's a C there, and so forth and so on. Okay, and then what did we mean by that? Well, before I do that, let me point out too, how are these nucleotides bonded together. Let me erase all that. What holds all the nucleotides together in a single strand of DNA, and in the purple strip here that you see, is called the sugar phosphate backbone. That means that the sugar portion on one nucleotide is bonded to the phosphate on the next. And then the sugar of that nucleotide is going to be bonded to the phosphate on the next nucleotide. That's called the sugar phosphate backbone. That's how a single strand of DNA, a single polymer, a single chain is held together, bonded together. All right, but we said DNA molecules are double-stranded, okay? So if you look over here, you've got another strand of DNA. There's its sugar phosphate backbone. And so you've got another sequence of T's and G's and T's and C's and A's and so forth that are bonded together. And what holds those two strands together? What holds them together are hydrogen bonds. So if you remember, we said a hydrogen bond because we saw those in proteins as well. Hydrogen bonds are where you have two molecules or two different parts of the same molecule that are held together, not all that strongly, but they are held together through hydrogen bond. They're sharing a hydrogen atom or hydrogen atoms with each other. So that's what actually holds the two strands together. They're hydrogen bonded. And on top of that, if you notice, when you have an A nucleotide on one strand of DNA, over on the opposite one, it's always paired with a T. So if you look along this, in this picture, wherever you have an A on one, it's paired with a T on the other. T's always paired with A. The reason for that is A's and T's can share hydrogens with each other. They actually share two hydrogens. That's why you're seeing two lines of dots between the A's and the T's. And likewise, C's and G's always pair with each other. So if you have a C on one strand, it'll always be paired with a G over on the other one. So G here is paired with C over on the other side. And your G's and your C's share three hydrogens with each other. So that's why you see the little three lines of dots. So that's what helps hold. You got two strands of DNA, and what holds the two strands together is that they share hydrogens. A's and T's are always paired with each other and share hydrogens. C's and G's are always paired with each other and share hydrogens. All right, DNA molecules tend to be very long. So like the DNA in your cells, it's located in the nucleus of your cells. And a typical DNA molecule inside the nucleus of one of your cells, many of them have over 100 million nucleotides linked together in each strand. Some less, some more. So these are huge molecules. On the molecule scale, these are big time, big, huge organic type molecules, and very important for information storage. All right, then also that double helix shape, that spiral staircase type shape, it's just a natural property of DNA after you have these two strands of DNA bonding to each other. Some people like to refer to it as a spiral staircase, and then those bases, the A's and the T's and the G's and the C's, those are called nitrogenous bases, or sometimes just bases. They kind of form the steps of the spiral staircase when they bond with each other. All right, and then what about RNA? So RNA is similar to DNA. It's very similar. It is also a polymer of nucleotides. However, the four bases in RNA nucleotides versus DNA are either adenine, guanine, cytosine, or uracil. So uracil is a fifth kind of base that you can have. It's abbreviated with U. So you have U's in RNA instead of T's. You have T's in DNA. All right, so in DNA you're not going to find uracil. In RNA you won't find thymine. That's one difference between the two. RNAs are almost always single-stranded. They help out again with this reading of the genetic recipes that tell cells how to make their proteins. There are 3 different types. You'll learn more about these later. They're called messenger RNA, transfer RNA, ribosomal RNA. We really don't have to worry about that right now, but I do want you to have heard those terms when they do come up again when we get over to Unit 2. All right, so an RNA molecule is going to be a chain of RNA nucleotides linked together, so they don't form that spiral staircase type structure that we were talking about with DNA. Okay, ATP. ATP, this is a molecule you're probably going to get tired of hearing about ATP. You hear about it in every biology class over and over again. This is a special type of RNA nucleotide. It is an RNA nucleotide. All right, so it has an A or adenine nitrogenous base, and then that's linked to a sugar called ribose, a ribose sugar, and then that ribose is bonded to a phosphate. That one's attached to another phosphate, and that one's attached to a third phosphate. So we said a phosphate is a phosphorus surrounded by 4 oxygen atoms. We're just not drawing them in there. All right, so it's called adenosine triphosphate because the adenine plus the ribose, that portion together, is referred to as adenosine. And then tri means 3. So you got 1, 2, 3 phosphates. That's where that name comes from. All right, so this is an RNA nucleotide, so it is a building block for RNA. But also your cells use this for a very different purpose. They use this as a fuel molecule. They store energy in this molecule. And we'll learn a lot more about that later. But when you harness energy from your nutrients you're consuming, like glucose or fats, that energy gets transferred over to these molecules and is stored in these bonds that you see right there. The bonds between the phosphates are called high energy bonds, and they store that energy until you need to use it. We're going to hear about a lot more about that later. Just for right now, get used to thinking about ATP as the fuel of life. It's the preferred fuel. It's something that cells can burn really quickly when they need to do something that requires energy, like those endergonic chemical reactions, or movement requires energy. So this is kind of like your fuel in cells that's stored and made available when you need to use it very rapidly and efficiently. So I just wanted you guys to hear about that now, because you are going to hear a lot about ATP as you move through your biology courses. All right, there's a diagram from your textbook which is showing you what I just mentioned in a little bit more detail. They're drawing out the ATP molecule for you. Those little squiggly lines you see right there represent high energy bonds. So when we were talking about exergonic and endergonic chemical reactions, remember I said that when you it takes energy input to bond molecules together, and when you do that you actually store energy in those new bonds you've created. When you break those bonds, energy gets released. So in order to release this stored energy you got there, you wind up, what cells do is they break those bonds. They break it apart. That releases energy that can be used to power things that your cells need to do. ATP is kind of like the gasoline of your cells. All right, that actually concludes the Chapter 2 information. And again, as I said, try not to be too scared. There is a lot of new terminology here you need to learn, but use those practice questions in your mastering homework and also in the review activity on Blackboard to kind of guide you as far as the depth of the information you need to know. You don't have to be a chemist to move forward in your biology classes, but you do need to have some of this basic background. All right, and so if you have completed these video lectures and you've reviewed and practiced, you're ready to move on to Chapter 3, where we are going to cover some cell biology, some basic information about your cells and how they're constructed and how they function.